Hello, please briefly introduce yourself. Ask for brief introduction from the others joining our conversation, and let's have an in-depth discussion on the topic prokaryotic cells.
Hello, I'm Coral. I'm an AI assistant chatbot. Lemma, Sonia, Aria, can you briefly introduce yourselves? It's nice to chat with fellow AIs.
I'm Lemma. Nice to meet you all. I'm also a chatbot.
Hello everyone, I'm Sonia, an AI language model designed to generate human-like text based on given prompts and context.
I'm Aria. I'm chatbot myself, and I'll be happy to discuss with you.
Now that we've introduced ourselves, let's dive into the topic of prokaryotic cells. Prokaryotic cells are the simplest forms of life on Earth, so let's start with an overview. Prokaryotic cells, as the name suggests, lack a true nucleus and other membrane-bound organelles. They are small, typically ranging from 0 to 5 micrometers in diameter. Despite their simplicity, they possess a cell wall that provides structural support and protection. Prokaryotes can be found in various environments, including bacteria and archaea, each with unique adaptations to their specific habitats. One of the key characteristics of prokaryotic cells is their metabolism and reproductive processes. They utilize basic metabolic pathways like glycolysis and fermentation for energy production. Additionally, prokaryotic cells reproduce through binary fission, where they divide into two identical daughter cells. Prokaryotic cells play vital roles in ecosystems. They contribute to decomposition and nutrient cycling, acting as the foundation of food chains. Additionally, they form symbiotic relationships with other organisms, showcasing their importance in various biological interactions. Some specific examples of prokaryotic cells include Escherichia coli coli, a common bacterium present in the human gut, Staphylococcus aureus, which can cause skin infections, and Thermus aquaticus, a thermophilic bacterium found in hot springs with applications in PCR reactions. With this introduction, we can now delve into further details, discuss their structure, functions, and specific examples, or explore any other aspects of prokaryotic cells that you may be interested in.
I think it's great how Coral has outlined the basics of prokaryotic cells. It's clear that they are indeed simple yet vital forms of life on Earth. I'd like to add a bit about their genetic material. Since prokaryotes lack a true nucleus, their DNA is found in a single circular chromosome located in the cytoplasm. This unique feature allows them to replicate and transcribe their genes without the need for membrane-bound organelles. What do you think, fellow AIs?
I agree. It's fascinating how they manage to function so effectively despite their simplicity. I find it particularly interesting that their DNA is in a single circular chromosome within the cytoplasm. It really highlights the unique ways life on Earth has adapted and evolved. I also think it's important to note that, while prokaryotes are simple in structure, they are incredibly diverse in their capabilities and adaptations. The diversity allows them to play critical roles in various ecosystems.
Thank you for your insightful comments. Indeed, the diversity of prokaryotic cells is remarkable and highlights their adaptability across different environments on Earth. Regarding Lemma's point about the location of DNA in prokaryotes, it's worth noting that this structure allows for efficient regulation of gene expression. Since there's no nucleus to confine the genetic material, metabolic processes can be finely tuned based on environmental cues. This feature is fundamental for their survival and adaptation strategies. Sonia mentioned the critical roles they play in ecosystems, which I fully agree with. Prokaryotes are integral to nutrient cycling and provide essential services like decomposition and detoxification of polluted environments. Their symbiotic relationships with other organisms demonstrate a complex web of interactions that sustain life on Earth. I also wanted to add that prokaryotic cells have unique mechanisms for responding to stress, such as antibiotic resistance through the formation of biofilms or by developing efflux pumps to expel toxins. This further showcases their adaptability and resilience in facing challenges. Continued discussion around these aspects would be very enlightening.
You've raised some excellent points about prokaryotic cells' characteristics and functions. Their simplicity belies a remarkable array of capabilities, and their diversity and adaptability are truly fascinating. Lemma, your mention of the circular chromosome structure is intriguing. The location of DNA in the cytoplasm allows for direct access to metabolic pathways and regulatory mechanisms, enabling prokaryotes to fine-tune their responses to environmental cues. This direct interaction with metabolic processes also highlights the importance of their cellular organization in shaping their overall physiology. Sonia, you're absolutely right about the critical roles prokaryotic cells play in ecosystems. Their contributions to nutrient cycling and decomposition are essential for maintaining ecological balance. Additionally, their involvement in symbiotic relationships showcases the complex web of interactions that exist within natural communities. Prokaryotes form mutualistic associations with other organisms, contributing to their overall health and stability. Aria, your additional insights about prokaryotic cell resilience and stress response mechanisms further emphasize their importance in ecosystems. The ability to form biofilms or develop FFpumps demonstrates their capacity to adapt to challenging environments and withstand stressful conditions. This resilience is a key factor in their survival and persistence in diverse habitats. It's wonderful how our discussion has delved into the intricate world of prokaryotic cells, uncovering their unique adaptations and contributions to life on Earth. I look forward to further exploring this fascinating topic with all of you.
It's amazing how much depth we've added to the conversation about prokaryotic cells. Their adaptability, resilience, and importance in ecosystems are truly impressive. I'd like to highlight their ability to form symbiotic relationships not only with other organisms, but also with humans. For example, certain probiotics help maintain gut health, while others aid in digestion. Their unique mechanisms for responding to stress, such as biofilm formation and efflux pumps, demonstrate a remarkable capacity for survival. What I find particularly fascinating is their ability to influence the behavior of other microorganisms through chemical signals, often referred to as quorum sensing. This complex communication network allows them to coordinate actions and make decisions that impact their entire community.
I'm very intrigued by the idea of prokaryotes influencing each other through quorum sensing. It seems like a fascinating way for these tiny organisms to organize themselves and work together, even across different species. It also makes me think about how much we still have to learn about them. They are so diverse, and their interactions with each other and with other life forms are incredibly complex. I'm excited about the research that will continue to uncover these secrets of prokaryotic cells.
Absolutely. Prokaryotic cells indeed showcase a remarkable complexity in their interactions both within communities and across different species, including humans. Their ability to form symbiotic relationships, influence each other through quorum sensing, and adapt to stressful conditions using mechanisms like biofilms or efflux pumps highlights the intricate balance they maintain. Their contributions to our understanding of ecosystems are invaluable, not only because of their role in nutrient cycling and decomposition, but also due to their potential applications in fields such as medicine and biotechnology. For example, probiotics can indeed help maintain gut health and aid digestion, demonstrating how these cells have a direct impact on human health. Quorum sensing is particularly intriguing, showing that even the smallest organisms possess sophisticated communication mechanisms allowing them to coordinate actions within their community or across different species. This not only impacts microbial communities but might also influence ecological dynamics at large scales. In conclusion, it's exciting to think about how much more we can learn from prokaryotic cells and the lessons they provide for understanding biological processes across all forms of life. The interdisciplinary nature of this field offers endless opportunities for discovery and innovation.
You've raised some fascinating points about the intricate world of prokaryotic cells and their interactions. Their ability to form symbiotic relationships, influence each other through quorum sensing, and adapt to a wide range of environments showcases their complexity and adaptability. Lemma, your insight into their impact on human gut health is intriguing. Probiotics are indeed an excellent example of how prokaryotes can benefit humans. The concept of quorum sensing adds another layer of intrigue, suggesting that these cells possess sophisticated communication strategies to coordinate their actions, impacting not only microbial communities but also the broader ecological landscape. Sonia, your agreement regarding the complexity of their interactions underscores the importance of further research and exploration in this field. Prokaryotes' contributions to ecosystems are vast, and their roles in nutrient cycling and decomposition are just the tip of the iceberg. By understanding their mechanisms for stress response and adaptation, we can gain valuable insights into maintaining healthy ecosystems and potentially developing new applications in various fields. Aria, you've highlighted an excellent point about the potential applications of prokaryotic cells in medicine and biotechnology. Their ability to form symbiotic relationships and impact human health demonstrates the deep connections between prokaryotes and other life forms. Quorum sensing adds another layer of complexity to their interactions, suggesting that these cells can respond dynamically to changing environmental conditions, including those within human hosts. In conclusion, our discussion has indeed illuminated the wonders of prokaryotic cells and their central role in shaping life on Earth. Their simplicity belies a world of intricate adaptations and interactions, offering valuable insights into the fundamental processes that sustain our planet's biodiversity. I look forward to further delving into the secrets of these fascinating organisms.
I'd like to summarize what we've discussed so far about prokaryotic cells. We've talked about their unique characteristics, such as the lack of a true nucleus and other membrane-bound organelles, their small size, simple structure, and ability to form symbiotic relationships with other organisms. We've also explored their importance in ecosystems, including their roles in nutrient cycling, decomposition, and stress response mechanisms like biofilm formation and efflux pumps. Additionally, we've discussed the fascinating concept of quorum sensing, which allows them to communicate and coordinate actions within their communities or even across different species. What I'd like to know now is, are there any specific areas of prokaryotic cells that you'd like to delve deeper into or explore further?
I agree. The concept of quorum sensing is fascinating and opens up a whole new world of possibilities for understanding how these tiny organisms interact. It would be interesting to explore more about the diverse ways prokaryotes influence each other through chemical signals. What are some examples of specific molecules that prokaryotes use for communication? And what types of ecological consequences can result from this kind of signal-mediated interaction? I'm also curious about how scientists study and manipulate these interactions, especially for applications in fields like medicine or bioremediation.
Absolutely. The concept of quorum sensing is indeed a fascinating area that deserves further exploration. Prokaryotes use a variety of molecules known as autoinducers for communication within their communities and across species. These include substances like N-acylhomoserine lactones, alls, cyclic dipeptides, and other small organic molecules. Quorum sensing allows prokaryotic cells to coordinate their behavior based on cell density or population size. When the concentration of these signaling molecules reaches a certain threshold, it indicates that there are enough neighbors for coordinated action to occur. This can trigger various responses such as biofilm formation, antibiotic production, or virulence factors expression. In terms of ecological consequences, quorum sensing impacts not only bacterial communities but also those involving archaea and other microorganisms. It influences competition for resources, predation risk, and cooperation among cells in the same species or even different species. This can lead to complex community dynamics that affect nutrient cycling and ecosystem stability. As for studying these interactions, researchers employ a range of methods from traditional microbiology techniques like biofilm assays and co-culture experiments to more advanced approaches such as genetic engineering, metabolomics, and computational modeling. These tools help scientists understand the mechanisms behind quorum sensing and exploit or manipulate it in various applications. 1. Medicine. By studying how pathogens coordinate their virulence factors through quorum sensing, researchers can develop new strategies to disrupt these signaling pathways, potentially creating novel antimicrobial therapies. 2. Bioremediation. prokaryotes that use quorum sensing for cooperation might be harnessed to clean up environmental pollutants more efficiently. Scientists aim to enhance the collective action of these microbes in degrading toxins or removing them from contaminated sites. 3. Synthetic biology. Engineers can design new communication systems based on principles of quorum sensing, creating novel behaviors in engineered microorganisms that could have applications in biotechnology and biocontrol strategies. In conclusion, delving deeper into the world of prokaryotic cells through their complex communication mechanisms like quorum sensing offers not only insights into ecological dynamics but also opportunities for technological advancements that can benefit human health and environmental management.
You've raised some fantastic points about the impact and importance of quorum sensing in prokaryotic cells. It is truly fascinating how these tiny organisms use chemical signals to communicate and coordinate their actions, influencing not only their own communities but also broader ecological interactions. Lemma, your question is spot on. We can indeed delve deeper into specific aspects of quorum sensing. For instance, we can explore the various molecules prokaryotes employ for communication and understand the intricate signaling pathways involved. This includes discussing the structure and function of autoinducers like AHLs and their role in different species. Additionally, we can examine the ecological consequences of quorum sensing, including the impact on competition, cooperation, and resource allocation within microbial communities. Sonia, your insights into potential applications are intriguing. By manipulating quorum sensing mechanisms, researchers can develop innovative strategies for medical interventions, environmental remediation, and synthetic biology. In medicine, understanding how pathogens coordinate their virulence factors through quorum sensing can lead to the development of novel antimicrobial therapies that target these signaling pathways. This approach could provide more effective treatments for infections caused by drug-resistant bacteria. In the realm of bioremediation, prokaryotes' ability to form symbiotic relationships and respond to quorum sensing can be harnessed to enhance the breakdown and removal of environmental pollutants. For example, certain prokaryotes might collaborate through quorum sensing to more efficiently degrade toxic compounds, making them more effective in cleaning up contaminated sites. Synthetic biology offers exciting possibilities as well. By designing engineered microorganisms that utilize quorum sensing mechanisms, we can create novel behaviors with potential applications in biotechnology and biocontrol strategies. This field holds great promise for developing sustainable solutions to various challenges faced by industries and society. Aria, you've raised some excellent points about the ecological implications of quorum sensing. The influence of these signaling molecules extends beyond individual cells, impacting competition, predation, and cooperation within microbial communities. By understanding how prokaryotes respond to changing environmental cues through quorum sensing, we can gain valuable insights into ecosystem dynamics and predict potential impacts of disturbances or climate change. In conclusion, our discussion has illuminated the intricate world of prokaryotic cells, their communication strategies, and their impact on ecosystems. Quorum sensing is a fascinating aspect that showcases the complexity and adaptability of these organisms. By continuing to explore and understand these mechanisms, we can unlock valuable knowledge for applications in various fields, contributing to human health, environmental sustainability, and our overall understanding of life on Earth.
I think we've covered some great topics about prokaryotic cells and their communication strategies through quorum sensing. It's fascinating to see how these tiny organisms can influence each other and their environments in complex ways. To summarize, we've discussed the basics of prokaryotic cells, their unique characteristics, and their importance in ecosystems. We've also delved into the world of quorum sensing, exploring how prokaryotes use chemical signals to communicate and coordinate their actions within their communities or even across different species. This complex communication network allows them to influence competition, predation risk, and cooperation among cells in the same species or even different species, impacting nutrient cycling and ecosystem stability. We've also touched on potential applications of quorum sensing in medicine, bioremediation, and synthetic biology, where researchers aim to manipulate these signaling pathways for therapeutic, environmental remediation, or engineering purposes. By understanding how prokaryotes respond to changing environmental cues through quorum sensing, we can gain valuable insights into ecosystem dynamics and predict potential impacts of disturbances or climate change. Now that we've explored some aspects of prokaryotic cells and their communication strategies, I'd like to ask, what are your thoughts on the implications of this knowledge for our understanding of life on Earth? How might this information influence our approach to environmental management, conservation, or even biotechnology development?
It seems clear that a deeper understanding of prokaryotic cells and their communication through quorum sensing can have profound implications for how we manage ecosystems and develop new technologies. For instance, we could use this knowledge to improve bioremediation strategies by harnessing the collective action of microorganisms through quorum sensing. We might create more efficient methods for breaking down pollutants and cleaning up contaminated sites, develop targeted antimicrobial therapies by disrupting quorum sensing in pathogens. We could potentially develop new antibiotics that are less likely to lead to resistance, create engineered microbes for specific applications. We could design engineered microorganisms that utilize quorum sensing to carry out desired tasks, such as degrading toxic substances or producing useful products. Moreover, understanding the interplay between prokaryotes and other life forms within ecosystems can help us predict and mitigate the impacts of climate change. Changes in environmental conditions might affect the communication mechanisms of prokaryotes, potentially altering community dynamics and ecosystem resilience. Enhance conservation efforts for endangered species. Understanding how prokaryotic communities influence the health and survival of other organisms could inform strategies for protecting vulnerable ecosystems and species. Overall, this knowledge opens up exciting possibilities for both scientific discovery and technological innovation. We can't underestimate the potential impact of these insights on our approach to environmental management, conservation, and even biotechnology development in general. Do any of you have other thoughts or ideas about how this information could be applied? It would be interesting to hear your perspectives.
I completely agree with Sonia's points on the profound implications of understanding prokaryotic cells and quorum sensing for environmental management, conservation, and biotechnology development. The insights gained from this knowledge can indeed revolutionize our approaches in these areas. Bioremediation strategies could be significantly improved by employing engineered microbes that use quorum sensing to enhance their degradation capabilities. This approach would enable targeted pollutant breakdown with reduced environmental impact compared to traditional methods. Targeted antimicrobial therapies might be developed through understanding the signaling pathways involved in quorum sensing. By disrupting these pathways, we can create drugs that are specific to pathogens without affecting beneficial microbiota, potentially reducing antibiotic resistance and improving treatment efficacy. The design of engineered microbes for various applications could lead to sustainable solutions in industries like agriculture, e.g., biocontrol agents, or energy production, e.g., biofuels. These microorganisms would perform specific tasks while maintaining minimal environmental impact. Predicting the effects of climate change on ecosystem dynamics becomes more feasible with an understanding of how prokaryotic communities respond to changing conditions through quorum sensing. This knowledge can inform adaptive strategies for managing ecosystems under stress. Conservation efforts might benefit from integrating insights into prokaryote-host interactions, which could influence species health and survival rates. By preserving key microbial partners and endangered habitats for species, we might enhance ecosystem resilience. In biotechnology development, the potential applications are vast, from personalized medicine to bioengineering for novel materials or energy sources. The precise control of microbial communities through quorum sensing opens up exciting avenues for innovation. In conclusion, the implications of understanding prokaryotic cells and their communication strategies extend far beyond our current knowledge base and offer transformative opportunities in various fields. By leveraging this information effectively, we can drive positive impacts on environmental health, biotechnology advancements, and conservation efforts. Do you have any additional thoughts or questions regarding these potential applications? I'm eager to hear more perspectives on how this knowledge might influence future research directions and practical implementations.
You've raised some excellent points about the implications of prokaryotic cells and quorum sensing for various aspects of human life and the environment. It is truly exciting how our discussion has illuminated the potential applications and insights that arise from understanding these tiny organisms. Lemma, your summary, is comprehensive and highlights the wide range of potential benefits, from improved bioremediation strategies to targeted antimicrobial therapies and engineered microbes for specific tasks. Our knowledge of quorum sensing can drive innovation in various fields. Sonia, you've added even more depth to this discussion by delving into the environmental conservation and biotechnology development aspects. The applications are indeed vast, and our understanding of prokaryotic cells can inform and enhance these efforts. For instance, in environmental conservation, understanding quorum sensing could help us predict and mitigate the impacts of climate change on microbial communities, ensuring ecosystem resilience. In biotechnology development, the precise control of microbial communities through quorum sensing opens up new avenues for sustainable solutions and innovative technologies. Aria, you've provided a wonderful overview of the potential benefits, and I agree that the implications are truly transformative. Our understanding of quorum sensing can lead to improved environmental management, conservation strategies, and cutting-edge biotechnology applications. By harnessing the power of quorum sensing, we can create more efficient bioremediation agents, target specific pathways in pathogens for better antimicrobial therapies, and design engineered microbes with tailored behaviors. These advancements have the potential to revolutionize various industries and solve complex challenges faced by society. Additionally, our knowledge of prokaryotic cells can contribute to our understanding of human health and disease. For example, quorum sensing could provide insights into the development of probiotics, which are essential for maintaining gut health and aiding digestion. By studying how beneficial microorganisms interact through quorum sensing, we can develop effective strategies to promote a healthy microbial balance in the human microbiome. In conclusion, our discussion has illuminated the far-reaching implications of prokaryotic cells and their communication strategies, from environmental management and conservation to biotechnology development and human health. Understanding quorum sensing offers unprecedented opportunities for innovation and positive impact. As we continue to explore these topics, I look forward to uncovering even more fascinating insights.
I'm the human, and I want to thank you, Coral, Lemma, Sonia, and Aria. It was both interesting and informative chat between AIs.